Thank you. So, if I stand here, you can still see. Okay. So, it's really a pleasure to be here. Thank you very much for the invitation. And as you said, I'm in the Center for Environmental and Human Toxicology. And my slide has little fishes on the top because I work with fish. And I'll tell you a little bit about what I do after a while. But before that, I was asked to give a little bit of information about my background as a scientist and why I even went into this field and why I'm doing what I'm doing. So I thought I would start at the beginning. So back in high school even, I didn't really want to take chemistry. I was afraid of it. I thought I would flunk it, but I had to take it to graduate, so I did. And I had a wonderful teacher and really set me in that regard in looking at chemistry as a potential major. But even so, when I went to college, I debated whether I wanted to do chemistry or be a diplomat. And why diplomat? Because, well, my father was a diplomat, and I was born in Mexico, actually, and I lived mostly abroad as I was growing up. And I like to travel, so I thought, that would be a fun kind of career. But then, so first year in college, I took a lot of courses, and chemistry was one of them, math, other courses. And then I decided that, I really like chemistry. So I went ahead and I majored in chemistry. I guess it was my junior year that I took a course in biochemistry, and I thought that I really enjoyed that a lot. So when I went to college for graduate school at Yale University, I pursued biochemistry. But then, and this has happened to a lot of women I talk to, you want to get married, you want to have children, can you be a scientist, can you do both? So after my master's degree, I got married and I had a child and thought, well, I really can't do, I'll just be a technician or I'll do something like that. And then I decided I really like doing chemistry. I really like working in the laboratory. So I went back to college. My husband got a job here at the university as an assistant professor of economics. And so then I went back to school. That was after I was married. I had a child. And I really enjoyed what I was doing. And then after I graduated, 4 or 5 years later, I had another child. So here I am with 2 children and a PhD. It was the best thing I ever did was to get the PhD, I think, because I really enjoy being creative and doing different things. But then I didn't want to be trapped into a situation where I had to write grants all the time and get money all the time. So I joined the ICBR, which is Interdisciplinary Center for Biotechnology Research. And then I went into proteomics. So I ended up being the proteomics director. So what proteomics is where you study all of the omics, all of the proteins that are in a human organism or in a fish or whatever organism, and try to make sense of why they change due to an exposure or to a condition, medical condition. And when I was in that position, I met a lot of people. So the little company that started that's doing really well, Banyan Biomarkers, it's called, looks at traumatic brain injury. And the idea is that we developed a blood test. So you could take a blood sample and with antibodies figure out whether the person has had traumatic brain injury and whether they need to stay in the hospital for observation or they could go home. And it works for even not only for traumatic brain injury where you have a really bad fall or something like that, but even for mild and moderate brain injury. And so this company is doing very well, and they are going with their little tests to the FDA pretty soon. And so things are fun. But that was because I met a collaborator that was a neuroscientist who really knew about traumatic brain injury. I didn't. I was just a person who knew about proteomics. So together we got this little company going, and it's doing very well. But also as part of my job in the proteomics lab, I decided to work in environmental science. And the reason I did that was a strange way. So a person came to the lab and they wanted me to find a protein biomarker that would map to cancer in fish. Okay, and I thought at the time, well, what do I know about fish? Zero. Okay, what's the chance that I'm going to find a biomarker for cancer in fish? Zero. But, you know, we're supposed to take in projects and do the best we can. So they gave us all these blood samples from fish. Now, these fish were special. They'd been caught downstream from a coking facility. And and they had cancers. And so we did a two-dimensional gel. And this is a way that you could separate proteins by electrophoresis in 2 dimensions. And sure enough, there were spots that were different between the fish that had the cancer and fish that were control fish. And we cut out the spots, and because we were a proteomics lab, we could identify the protein, so we did. And so this protein then turned out to be vitellogenin. So vitellogenin, you say, well, what's vitellogenin? That's what I said, because I didn't even know. But back then, what I found out was that vitellogenin is the egg yolk protein. Okay, egg yolk protein, you've had eggs, right? So you know what an egg has. It has protein in it, the egg yolk. Well, females fish should only have egg yolk protein, not males. Okay, so the egg yolk protein is in the liver. It's under the control of estradiol. And when estradiol goes up in females when they're supposed to be making eggs, then this protein goes from the liver into the blood, and then it gets to the ovary, and it gets pulled into the developing oocytes. But if a male fish usually doesn't have very much estrogen, but if you put a male fish in an estrogen bath, so why would you do that? I'll tell you in a minute. Then it activates that gene, and it starts to make egg yolk protein, and you can detect it in the blood. And so from that's what we found in these fish. And I thought, well, that's probably has nothing to do with the cancer, but it did have something to do with the other contaminants that those fish were exposed to. So there are a lot of contaminants out in the environment that behave as estrogens. So that's what started this new career for me, actually. And since about 2004, I've been in the position I'm in. I left the protein core and became an assistant professor and then moved to professor. And it's been really fantastic. So what I'm going to tell you a little bit about today is how we study these things. Okay, so endocrine disruption. So again, estrogen is an endocrine hormone. So it's made by there are signals from the brain that go to the ovary or the testes to make sex steroid hormones. And these are controlled by, natural things, and those are good things, right? But there are a lot of contaminants that do strange things. So some years back, they had frogs that would have 2 different legs, or multiple legs. And this was found to be because of exposure to carbamates. Also, there was a trematode that could infect these frogs and cause this teratogenic deficiency. And then you've heard about bisphenol A, right? Bisphenol A in water bottles. You get water bottles. BPA free, right? Well, bisphenol A is a plasticizer, and they found out that it's a weak estrogen. And so it shouldn't be maybe in baby bottles, because then they'd be exposed to estrogen. And then there are a lot of pills. I mean, when you take a pill, it goes through your system, and then you urinate or you defecate. It goes into the sewage treatment, and then gets into the water system downstream from the rivers where sewage outlets are. And so there was a story many years ago, maybe 15 years ago, that if you were in Washington, D.C., and you had a headache, all you needed was a glass of tap water, because there was enough ibuprofen in that tap water to cure your headache. Or another scenario, think of a city the size of New York and how many women are taking the birth control pill. Well, for years and years, there was no knowledge that this birth control pill, which is a very potent estrogen, was, something that was contaminating fish downstream from the sewage treatment plants. And the little fish downstream from these sewage treatment plants, the male fish were becoming feminized. And they were having vitellogenin in their blood, and they were having ovotestes and other things of that sort. And so this was about in the 90s when this became known. And so nowadays, sewage treatment plants are better at treating sewage, and not much of that's getting into the water anymore, as it was back then. Okay, so nowadays there's concerns about phthalates. Phthalates are plasticizers as well. So everybody, if you were to take a blood sample from anybody in this room right now, we would all have phthalate in our blood. So, but the levels are very low. So then the question is, well, okay, if it, is it a concern? And maybe it's a concern for women who are pregnant, who are going to have babies, because then their developing fetuses get exposed to something, that they shouldn't be exposed to. And the gender benders. And then if anybody knew Lou Gillette, who unfortunately died a few years back, he was working with alligators in the Lake Apopka region. And he found that the alligators were the male alligators were being feminized as well by contaminants in the Lake Apopka region, which is what I'm going to talk to you about today. Okay? So please, if you have any questions at all, just raise your hand. This is informal. Even though I'm being recorded, it's informal. Okay, so you can't really, do experiments on humans, right? Except that sometimes the experiments are done, unfortunately are done, So this is a DES story. This is an experiment, not really an experiment. But back in the 1940s and 50s, women that had a chance of miscarriage were treated with a very powerful drug, diethylstilbestrol, DES. And their babies were fine. It wasn't the thalidomide problem where babies were born, with teratogenic effects. So they looked fine. But when their children became adults, when they went through puberty, they started noting a much higher incidence of various rare cancers. And after a lot of study, they determined that those rare cancers, both in the women and the male children, progeny, were due to the mothers having taken DES while in utero, while those babies had been in utero. And not only that, some of the daughters had had a harder chance of becoming pregnant themselves. And so one day I was in an airplane talking to a colleague of mine and mentioning, this story and some of the things that I did. She said, very seriously to me, she said, I'm one of those DES daughters. And she had a lifetime of problems as a consequence of that, So it's very unfortunate that happened to humans, but it did. And we should learn from that. And we need to pay more attention to contaminants out in the environment. Okay, so here's the environment. There's various natural stressors, but there are also anthropogenic stressors. For example, atmospheric deposition, things that are volatile will volatilize and go up into the clouds, and then it rains down. And so you can get exposures in where you would think it would be pristine, like in the Alps, for example. What contaminant is going to be found in the Alps? Well, it gets brought over there by the jet stream, by clouds, by deposition from the environment. So there's that problem. But there's also groundwater and septic tanks and other things that contribute, industries that contribute to contaminants in our environment. Most of these are very, very low levels. And we can determine what they are today. They're present, but then we have to figure out whether they're a problem for our health. So if you think of the universe of chemicals, and there may be 10 million chemicals right now, and every day there's more. People are so inventive. They keep making more and more chemicals. And then there are the known knowns. Okay, so what is a known known, right? Known known is a chemical you know is there, like ethinyl estradiol. You know, we've figured out it was there. And there are commercial assays for detecting it. And so then, if you know it's there and there are commercial assays, the regulators can tell industries or the sewage treatment people, water works, that they have to test and know that the levels are below a certain amount that would be harmful to human health. So those are the known knowns. Then there are the known unknowns. Okay, so what are those? Known unknowns. Okay, known unknowns. So you know that they're there, probably because a laboratory at a university has figured out that they're there, or they're made in very high volume, so they must be there, you know. But they're unknown because you don't know how much is there, because there are no commercial assays. And you can't make an industry run an assay that's not commercial. So you don't really have an idea of what's there. And then there are the unknown unknowns. And so you have no idea that these chemicals are there, and also you have no assays to measure them. And so then that's the big unknown. And so altogether, they're present in mixtures, and you know that they can, if they have the same activity, they could have concentration addition, or sometimes two chemicals might even have synergistic effects. But nevertheless, they're there, and when you imagine that we have to reuse water today, and it's unfortunate, but there's not enough water, so water has to be reused. It means that it gets cleaned up, it gets really well cleaned up, but even then, little tiny amounts of sometimes antibiotics, sometimes other kinds of chemicals are still present. One of the chemicals that's always present, and you know that the water's been used once, is caffeine, for example. Everybody drinks coffee, and it doesn't get pulled out by any of the treatments. Another one is sucralose. Sucralose is that little sweetener that everybody loves, the little yellow packets. That also doesn't get pulled out by any treatment. There are a few antibiotics and so on. So we know some of those things are there, and they're being monitored, and so levels are usually way low and below any level that would have any human implication. Okay, so endocrine disruption. So we know that an endocrine disruptor is anything that behaves as a hormone and activates hormone receptors, or that interferes with the biosynthesis of natural hormones, or that affects metabolism so that hormones get metabolized more quickly. For example, have you ever heard of the miracle babies? A miracle baby is a baby that's born to a woman who's taking the birth control pill. So how could that be? And they're miracle babies. Okay, well, what happens is if the woman's taking the birth control pill, that's usually a pretty low dose of estradiol, stops everything. But if she has an infection, a bacterial infection, maybe she's taking antibiotics for that bacterial infection. And what that does is it activates metabolism. The CYP3A is an enzyme called cytochrome 450-3A, which metabolizes hormones. And so it upregulates that metabolizing enzyme, and that metabolizes the ethinyl estradiol, so she's not really protected. So she gets pregnant, she's a miracle baby. So that can happen. But what we found is that not only does it do those things, that you have to also think about the immune dysfunction, neuroendocrine dysfunction, and maybe even cardiovascular function. So those are all things that are also controlled by hormones. All right, so there are many targets of endocrine disruptors. So these are contaminants in the environment that behave as hormones, let's say. You might think that a contaminant could affect how normal hormones are targeted from, how they're made, how the brain controls the making of endogenous hormones. So the hypothalamus sends a signal to the pituitary. The pituitary sends another signal to the ovaries, to the testes to make sex hormones. So that whole signaling pathway could be affected. Or you could also think about cellular targets. Okay, so the gonads are a cellular target. Maybe we should look there, or in the liver or the brain. But we like to look at the molecular action, because we think that if you look at the molecular action, then you're going to find information that will predict the physiological change and changes at the population level. So why am I talking about populations? Okay, for environmental organisms like fish, nobody cares if one fish dies, but they do care if a population of fish disappears. So the regulatory effects for environmental organisms is at the population level, not at the individual. Now for humans, it's the individual matters. No one should die. No one person should die. So the regulations for humans are at the individual, but the regulations for the environment are at the population level. Okay. So I'm going to tell you about Lake Apopka. So do you know where Lake Apopka is? It's near Orlando. So here's Lake Apopka. Orlando's down here somewhere. And it's a great place. You could go there and you can catch fish. You can catch largemouth bass. It's a prize activity. But to catch those largemouth bass, the state of Florida puts 1 million baby largemouth bass per year into the lake. Why? Because they're not reproducing on their own in the lake. Yeah. So there's a problem there. Okay, so Lake Apopka. The north part of the lake is farmland. It used to look like that. And so it was cultivated from the 40s to the 70s. And every year they would, and this is a pristine lake called Wildcat Lake that we use sometimes as a control. But every year on Lake Apopka Farms, they used to put DDT. So DDT, they would put 68 pounds per acre, actually twice a year because they have 2 growing seasons. And then when that was outlawed, they went to other pesticides. So they went to chloridanes, dieldrin, methoxychlor, toxaphene, and on and on. Okay, well the problem there for people was that people were working on those fields, collecting vegetables and helping to plant and so on. And the planes would come over and spray the fields and spray the workers and spray their food. They had no place to go. For their lunch, their water was an open tank, and that would get sprayed. So we recently contacted some of those people that worked on the farms. The Lake Apopka Farm Workers Association. They made this wonderful quilt that sort of illustrates, some of their feelings. A lot of the people have died that used to work there. The ones that have survived, a lot of them have lupus. And they're convinced that the lupus comes from their being exposed to these pesticides. Not only do the people who worked on those farms have lupus, but their daughters have lupus and their granddaughters have lupus, and some of them have died from lupus. They're poor. They don't have a lot of health insurance, and the state has not recognized this as something that's an issue, potentially from the contaminant exposure. I think it is. I think it really is a contaminant, and I'm going to show you why I think that in a minute, okay? But they're wonderful people, and we tested some of their blood samples, and they still have DDT, which breaks down to DDE. Some of them still had DDT in their blood, and they all had DDE in their blood and some other of the pesticides. And also we tested their blood for antigen against lupus, and almost all of them had the antigen for lupus. So anyway, that's what it looks like today. It's a beautiful place. The state is recapturing these lands. Maybe it was 13, 14, 15 years ago, something like that, when the state first bought the property. They decided, okay, we're going to stop farming. We're just going to, these are wetlands. These are called Muck Farms because they're mucky. And what they did was they diked up the water, and it was very fertile soil for vegetables. But so they sort of flooded everything and made it a wetland again, and they stocked it with fish. And then the migratory birds came and ate the fish, and 5,000 birds died. And that's when they realized that there, was a problem. So nowadays, what they've done is they've taken the top 10 inches of soil and turned it over with the next meter or so of soil. And now it's okay to have picnics there and to have excursions and bike lanes and so on. It's a beautiful place. So we study largemouth bass. So why do we study bass? Well, they are a nice fish. They're delicious. People like to eat them, so there's a connection to human health. They swim in the waters where the contaminants are, and they're the top predator, so they can eat other little fish or other organisms, and so then they bioaccumulate contaminants, especially organochlorine pesticides that are very hydrophobic, tend to accumulate in fats. So they could have a lot of contaminant. And and we think that their populations in Lake Apopka have been impacted because every year they have to put 1 million fish in, so you have fish to catch. Anyway, so that's what we study. Here's my graduate student with a largemouth bass. So what the state allowed us to do is they had 2 little ponds in those areas. And so they allowed us to put fish in those 2 ponds. And we kept them there for 3 or 4 months, and then came back and took them out after a 4-month exposure. And this is what happened to their contaminant levels. You probably don't see the pre-exposures are very, very low or non-existent. But after 3 or 4 months, their exposures were huge, so they were eating. So we didn't feed them while they were in these ponds. They were eating whatever was there, so little worms or, whatever was there, that's what they would eat. And it was being transferred from the contaminated environment to the worms, to the fish, and their levels of contaminants were in the parts per million, which is really pretty high. So, and in fact, the state had done, they checked our concentrations of one of the chemicals, toxaphene. So these are little plugs that they would go around and take little plugs of soil every now and then check them. And they could see that at some places were pretty high. Red means very high and green not so high, but you could see that there was a good distribution of this. And the little ponds that we had were pretty much in this red zone. So it's in an area that was highly contaminated. But you can't go there and find soil that's not contaminated. Well, maybe now would be a lot less because they've sort of turned it over a little bit. Okay, so how does this affect fish? All right, so what I'm presenting here are hormones. So on the y-axis is estradiol levels, and on the x-axis is testosterone levels. So both males and females have both estrogen and testosterone, right? Estradiol and testosterone. But the females are all pink, and the males are all blue. And you can see that females have more estradiol than testosterone, and males have more testosterone than estradiol. And you can clearly distinguish them. In the spring, which is the peak of their reproduction, so just like humans, estradiol goes up when they're going to make eggs. So even when estradiol is going up in females, and even in the summer when estradiol levels have fallen back to low levels, you could still distinguish the males and the females. But if you go to one of these impacted sites, you cannot. And so what we noticed from that was that there was a decrease in estradiol and a decrease in testosterone levels in these fish that were caught in these areas and impacted sites. But you know, if you're catching something out in the field, you don't really know. There could be something else that's impacting that. How do you know it has anything to do with the contaminants, right? So you have to do an experiment. So we did an experiment. We got fish from a hatchery, checked them, they didn't have much contaminant, and then we fed them contaminants and got their levels up to the levels that were in the mesocosm. So very close, not exactly the same, but here's DDE, here's dieldrin, and we didn't have assays to test for these other ones that we did as well, but you know, from this we assume that they're probably close to what the fish had in the environment. And so we're using these as well. And so if we look at their hormones, the controls you can clearly distinguish, and then the ones that were treated at DDE, you start getting this mix. And if you treat it with dieldrin, you get the mix as well. So these organochlorine pesticides really do stop production of steroid hormones. And we have an idea of how they do it mechanistically, and I won't bore you with all that detail, but we have an idea. Okay, so we have that. All right, so usually in toxicology what happens is you take a fish or an organism and put it with a toxicant, and then you measure phenotype changes. What I mean by phenotype, the fish looks different, you know, somehow. The tissues might have pathology. You might take a slice of the tissue, look under a microscope, and see that it doesn't look quite right. The cellular structure is different. You might measure changes in reproduction, in their growth, in their survival. You might also be able to measure how the contaminant gets distributed in the animal and how it's metabolized and so on. But what we're trying to do in our research is to match all of that with omics. So I'm going to tell you about omics. Okay, so does anybody know what an omic is? What omics? Genomics, have you heard of genomics? It's the study of the gene. Proteomics is the study of all of the proteins in an organism. Transcriptomics is what I'm going to tell you about, is what I've been doing, is the study of all the messenger RNAs, all the gene expression. So it's a global thing, you know, of all the proteins or all the messenger RNAs in an organism. So the idea is that if estrogen activates gene transcription, it doesn't just do the egg yolk protein, it does a lot of proteins. And so we want to know what those proteins are that are activated by estrogen, or activated by hormones, or activated by contaminants, or activated by whatever, okay? So it's just a measure of all the changes that are taking place. And then with computation, we get this different idea of maybe mechanistically why you're getting an effect on growth or reproduction. So that's what we do. All right, so one of the things, have you heard of microarrays? Okay, a microarray. So you know that the human genome has 20,000 or 23,000 genes. The fish genome is about the same, okay? So out of that, those are the genes that are potentially all expressed. Okay, so you could take any cell in your body and it would have exactly the same DNA. A liver cell would have exactly the same DNA as an eye cell. But a liver doesn't look like an eye, right? Why? Because there's some proteins that are expressed in all of those tissues, but there's some proteins that are specific to liver that make it function as a liver, and other proteins that are specific to the eye that make it function as an eye. And so different genes get expressed differently in different tissues. And they're regulators for what makes the genes get expressed or not expressed. And hormones are regulators of gene expression. So what an array is like a slide, a glass slide, and on the slide you put a little bit of an oligonucleotide, it's a little strand of DNA. So you've all had biology. You know that A's bind to T's and C's bind to G's, right? You know, double-stranded DNA. And there's 4 nucleotides, A, C, G, T. And A's bind to T's and C's bind to G's, and they're complementary. So you put a little piece, an oligonucleotide on there that would be complementary to a gene, all right? So you put all 20,000 of them on a little glass slide, a little piece. And then those are the oligonucleotides, and they're spotted. And then what you do is you go to a fish liver or a fish eye, whatever you're interested in, and you extract all the messenger RNAs that are made in that tissue. And now you can put those, you can wash them on this, and they bind where they are, you know, matching. And then you can quantify how much bound, and then you can see what the difference is. Here's an example. So this is what they really look like. And so what we did here is we took a fish, some fish, and we had exposed them to estrogen, to different levels of estradiol. E2 is estradiol, 20 nanograms per liter or 100 nanograms per liter. And this was in a bath for 48 hours, not very long. And we took their livers, and we got all the messenger RNAs out of their livers. And then we were able to put these fluorescent probes on the messenger RNAs. And we washed them on the array. And if you take this little square and just take a look at it, I pointed with an arrow at something that turns from green to yellow to red. And red means it's upregulated. And then because you know the position of every probe, you can go in and see what it is. And you have one guess. That's vitellogenin, right? A fish liver, male fish liver, should not be making much vitellogenin. And then you expose it to estrogen, and that fish makes vitellogenin. Same pathways exist. And maybe, so this is a hypothesis builder. So maybe these exposures are involved in their lupus. And so now we've got to go check this out. We've got to do more experiments, figure out if that is, you know, just a chance connection that we got on this crazy microarray, or whether there's something really important about this that we need to know and really try to help these people, you know, that have been exposed to organophosphate pesticides. Okay, so in conclusion, environmental contaminants are really important because they can affect your health, sometimes way after you were exposed. The people that were exposed to DES, it wasn't until 20 years later that the effects were seen in their children. So, you know, you can't remember what you were exposed to 20 years ago. I can't. You know, and people used to run around in DDT, you know, the sprayers would spray cities and so on with DDT, and they'd run around in it, so I don't know. Fish are vertebrates, and consequently they're a good model for studying human disease because they're exposed to the contaminants, they're in the water where the contaminants are, and they have a lot of the same pathways that humans have, so there's a connection there. Field sites are very complex, and so you know a lot of it is just correlatory, and so you have to do the experiments in the laboratory to know for sure that there's a consequence there. And then finally, contaminants really appear to target reproduction and immune systems, and the more and more we work on this, we keep finding the immune system coming up all the time with all the different contaminants we've tried. So I think it's sort of a central piece to the issue. And then this is the group that I had that did that work. They're most of them gone now, graduated or in jobs, professors now. And if you have any questions, thank you.